My name is Lyndel Meinhardt. I'm the research leader for the Sustainable Perennial Crops Lab, and thank you for having me. It's always a pleasure to join the HCP, and especially when we're presenting a new heirloom for the group. As I always do, I'm a federal employee. I work for the Department of Agriculture. I'm an agriculture research service scientist. I have to give a small disclaimer. Anything I mention here about types we are not allowed to endorse or promote any particular product over another. So just to be aware, I'm not promoting or endorsing, even though this may be the best HCP out there yet. I'm not allowed to endorse in any way, shape, or form. So, okay, let's get right into it. This is HCP 17. This is one of our newest ones. It's out of Guatemala. I think you've all have heard that now. I'm going to jump directly into the genetics of it. What we do is when the panel has designated this as an heirloom quality material, then we get material, we get the seeds now, and we can do analysis on the seed coat. So we basically take a small piece of the seed coat, we can extract the DNA from that, then we'll actually analyze that based upon particular genetic markers. And these are typically, they're called single nucleotide polymorphisms. And every individual on the planet, human, plant, whatever, has hundreds of thousands of differences that make us very unique. So we can actually identify an individual within a population based upon its marker profile. And what we've done, if you look at, for example, with domesticated dogs, for example, we have all the different breeds. They have markers that can identify each individual breed that's there and individuals within a particular breed with these markers. We can do the exact same thing with tree crop, and Theobroma cacao is how we're doing that. So we're actually able to take a bean, take the seed coat from it, extract the DNA, and we can tell you the genetic diversity of the mother tree that came from, because the seed coat is primarily the same genetic makeup as the mother tree, which means it's clonal with the mother tree. So the seed coat will tell you exactly what the mother tree is. What we've found is we've looked at a number of individual beans based upon characteristics of the bean. We try to separate the beans by eye, make sure that we have all the different sizes, shapes, and just as a precaution that we're not only looking at just the one set of beans. And when we look at that, we actually are analyzing this. I believe these were analyzed with over 300 or 400 markers. And based upon what we're finding with that is that we're finding that we're able to separate these into larger groups. And what we've done is with the larger SNP marker base that we're a grouping of markers that we're using now, we're at probably about 14 population groups. And based upon that, we can then separate our individual beans from the HCP 17 into various groups. And I believe this is just the raw data showing how each one of these are separated into the population groups that we're looking at. If you can go to the next slide. This makes it a little bit easier to take that data that you saw in the blue screen, the data, raw data, and we can actually separate it and take a more of an image analysis of it and say, okay, this is separating it into pie charts. And what we found with the ones that we've analyzed, we can separate our HCP 17 into 4 main groups. And what we see is that we have a number of trees that are the Trinitario Scavina 6 mixture, which is listed as Trinitario Ska. We also have a straight Trinitarios, which are a mixture between the Criollo and the Melanado. So what we have when we have a Trinitario Ska mix in the first group I mentioned, we have a Melanado population group, we have the Criollo population group, and we have this Ska 6 or Ska population that was found many years ago in Peru. And so those 3 are basically almost in equal portions in that group. We also have a Trinitario Nacional mix, which is more on the Criollo side, a larger percentage of National with a smaller percentage of the Amelinado, which would indicate that this is a cross between National and a Trinitario. And finally, we have a kind of unique group that we've not yet seen in any of our previous HCPs, is that we have a Trinitario Jurura cross. And Jurura is a river that we've identified a new population on. It's out of Brazil, but it actually extends somewhat into Peru. And we do not know where this cross would have came from, except material that was collected along the Ucayali River. They may have accidentally collected some Jurura material that was then taken back and was somehow crossed with a Trinitario down the road. And they were considering it as a different population, but we've identified the Jurura as a separate population group. So the HCP 17 is made up of these 4 main groups of trees, and that's a good indication that you're probably picking up some unique flavor profiles based upon this unique mixture. We can go to the next slide, please. And finally, we always like to look at the structural chart, which we are showing the individual populations. As you go across here, you can see the Melinado and the Criollo, the IMC, and Nene, all of these groups, National, you can see them separated into a different color group. And then if you go over to the tight group of HCP 17, we see that there's a Criollo in almost every single group. There's a little bit of the IMC mixed in a few of them. There's also some National in a number of them, but there's also, and then you can see the Melinado. So this is just another way of looking at it, and we pack them together, and we're looking at the individual bar structures. Can we go back to that past the previous slide? That structure bar is basically exactly what we see down here in the lower right-hand corner of this. We can break them into the percentages of the different mixes within them, and so you can look at it as an individual bar and say this is a structure, the composition of that population group based upon the contribution from each one of these other family or population groups. So that basically tells us. It gives us an idea of what we have, and it also gives us a reference point down the road to say, this is the makeup of HCP 17, and this is, well, we can always go back and say, this is the material that was designated as HCP 17 and is responsible for the unique flavor profile that it has. Yeah, and so that brings the genetic diversity portion of the talk to the end. I hope that you all appreciate this, and I hope you enjoy the tasting section of it and can enjoy this new HCP 17. Thank you.